Hi everyone, thank you so much for joining our second webinar of the year. I'm just going to start off with a few bits of information before I introduce the speaker. So please note that this webinar is being recorded today and we will disseminate the link shortly afterwards for everybody that has registered. We're also really pleased to let you know that this is a CPD certified event, and you can expect to receive your certificates around a week after the session closes today. I'm obligated to say that the views expressed in the webinar are those of the authors only and do not necessarily reflect the views of EMQN. There is the option to ask questions today, so please feel free to submit any questions that you have during the session using the Q&A box in the banner at the bottom of your screen. There is an option to submit questions anonymously, so please don't feel shy about submitting questions. No question is too small or too big, so please feel free to submit them using the Q&A box. So, it's my sincere pleasure to introduce our speakers for today, Dr. Antony Adamson. Antony leads the Genome Editing Unit Technology Platform at the University of Manchester here in the UK. And I don't want to detract too much from his talk, but since 2018, Antony's worked to develop this facility into the hub of genome engineering expertise that it is today. The unit works with academics and industry partners to apply cutting-edge genetic manipulation techniques to generate bespoke model systems which serve to facilitate understanding of complex biological systems and disease pathology. Antony and his colleagues increasingly find themselves called upon to integrate new and ever complex CRISPR technologies to develop model systems of human disease, and have recently begun exploring the utility of CRISPR in diagnostics. As a former collaborator of mine and now a firm friend, I know that Antony is extremely knowledgeable about all aspects of genetic engineering and its application. He has an unparalleled enthusiasm for this rapidly evolving area of science, and I know he'll be more than happy to answer any questions that you have today. So without further ado, I'll hand over to you, Antony.
Okay, just start my video again. Hello, and thank you for the introduction there, Rachel. I'll just start my screen share. It's a pleasure to speak to you all today. Mainly gives me the opportunity to talk about CRISPR-Cas9, which is something of a minor obsession of mine. And apologies for the lights that's just started streaming through my office window with these rather inefficient blinds that I have. But today I'm going to break my talk down into 3 different areas. First of all, talk about modelling disease, which is what we do as a facility day in, day out. Secondly, we're going to talk about utilising some of those technologies for diagnosing disease as well. And then the final part of the talk, I'll move on to speaking about treating disease, which is not necessarily something we do ourselves currently, but it's really a very exciting move, this particular technology. So I've got a vague understanding of what EMQN does, so I thought I'd try and frame the talk today from the perspective of genetics and disease. So let's imagine a situation where we're doing lots of sequencing these days. We're starting to understand our genomes in a lot more detail through GWAS analysis, projects like the 100,000 Genomes projects, doing whole genome sequencing of families that present at clinic. And we may get a doctor or a clinician colleague come to us and say, we've identified a bunch of variants that are in this family, and we think that's what's causing this particular disease. But right now, we don't know for certain if they have actual functional role there. So they're variants of unknown significance. So asking the question, is this variant really important? And that's where we come in. We create experimental models of disease. We use an awful lot of techniques to do this. The primary technique these days is CRISPR-Cas9. We can do this in vitro and we can do this in vivo as well in lots of different experimental models. And then once we've produced that model system, the group can go away and use it in all different kinds of assays. But this is really what we do. We create those experimental models of disease. And this slide here gives a bit of a timeline. minimize this. This slide here gives a bit of a timeline of the evolution of the facility, and you can see how this growth in this technology has really catalyzed our development. So we look back about 10 years ago, we were really solely focused on transgenic mouse models, and the facility only really brought in other people's mice into the university. Then we started making mice the classic way, where you essentially shoehorn a bit of DNA into the mouse genome in a random fashion, essentially. And people have been doing this since about 1975 or so. And that was the kind of GM technique of choice there. I joined the facility towards the end of 2014, and my background as a molecular biologist, my remit was, can you get CRISPR-Cas9 working and start to make mouse models using this technology instead? So we started to integrate CRISPR-Cas9. It worked straight away, and we have never looked back. We gained a bit of a reputation, and we kind of started a bit of a CRISPR consultancy. So all these other people that worked in cultured cells would get in touch with us and say, hey, can you help us design a CRISPR-based manipulation or perturbation of the cell lines? We grew and grew and grew, added more people to the facility as the demand started to grow. Then I became the manager of the facility in around about 2018 or so, and recognizing there was this very, very clear demand for gene-edited cell lines, we rebranded the facility. It's the Genome Editing Unit. We added the manipulation of cultured cells into our service portfolio as well. We also started adding in some of the derivative CRISPR technologies like CRISPR interference and activation and base editing. And we've continued to grow since then, adding a further person in the last couple of years and started working with our local fly facility to make genetically modified fruit flies as well. And then by the end of this year, we'll be adding 2 more people. And one in particular will expand our model capability even further by starting to develop induced pluripotent stem cells. So both creating those stem cells and also genetically modifying them as well. So this sentence here at the bottom summarizes what we do. We design and build bespoke experimental models in collaboration with our colleagues. And just a quick note on that assay aspect I talked about earlier on. We work very, very closely with our other technology platforms in Manchester, of which there are many. So we're making these models. It may be a gene knockout. It may be one of those mutations that we're putting in the genome to mimic human disease. Maybe we're tagging a gene with something functional, or maybe we're deliberately overexpressing a gene. And at the end point, we can maybe work with our genomics core facility to detect omic changes, so things like RNA-seq for transcriptomics. Maybe there's changes in the way the chromatin structure is, and using ATAC-seq and this kind of thing. We work very closely with the flow cytometry and also our microscopy core facilities, bioimaging. Maybe we're putting on one of these tags here. It could be GFP or another fluorescent or luminescent reporter that perhaps we're labeling a particular type of cell in a mouse so that flow cytometry can enrich for that cell type. Maybe the cell's being used for time-lapse imaging to see how proteins change in terms of secretion or localization over time. Or we may work with our mass spectrometry core facility to map the interactome of cells once we, say, knock a gene out or put a tag on for proximity labeling, that kind of thing. I think it's worthwhile pointing out some of you will have come from academic laboratories. The kind of traditional view of core facilities is a kind of siloed research facility with a bunch of equipment you can book on and go and use that's maintained by someone. Whereas really the modern view, I would argue these days, is we're collaborators. We work with the research groups and design and tailor what we can do with those research groups and have a strategic and intellectual input on their research projects. But we also work with those other core facilities to develop these cross-cutting and joined up workflows that take you from generating the model in the first instance to generate the data at the end of that pipeline. But now I'm going to focus on CRISPR-Cas9 and why CRISPR-Cas9 has become the tool of choice for generating our model systems. So I mentioned the classic exogenous expression method. So this is a bit, let's return to that example of a clinician's got in touch and says, we found this mutation, this genetic glitch in a group of patients, help us understand it. So what we might have done in the past is generate a plasmid or a viral vector or some kind of construct like that we could deliver into our model system to start producing the mutant protein. So you can see here we've got the background wild type protein here, and this one's got this red line indicating the point mutation or something like that. And then they could use the cell or the mouse or the zebrafish, whatever model they're working with, to look at the function, how the function changes. It could be looking at protein stability, secretion, the interactome, gene expression. Completely depends on what the gene of interest normally does. But there are some problems with this setup. So, for example, normally genes are found on chromosomes, and they've got a very kind of gene-specific regulation. So you've got exon, intron structures with untranslated regions. You have promoters. You have enhancer regions. You have sequences that respond to stimulation, maybe sequences that respond to time of day. So by expressing from these small constructs, we've completely removed the gene of interest from its normal regulatory expression. Typically, we end up with too much protein as well, so that's another perturbation there. And let's not forget, the cell's got its own gene, its own version of the gene. That's being produced in the background. That normal protein, it may get in the way of our assay. So this is a really useful technology and approach, but whenever we generate data in this fashion, we have to recognize that the model we've generated has got some unintended perturbations. And maybe any conclusion we draw from this model, we maybe have to temper them slightly and say we have to accept and acknowledge there may be flaws in the model system we've developed. Whereas with gene editing, and there's been lots of gene editing techniques over the years, but CRISPR-Cas9 has become the default. So it's by far the easiest to apply. It's represented here by these molecular scissors, if you will. What we can do is take that same mutation, but program it directly into the endogenous gene within the genome. What this means is the gene is still controlled by its normal regulatory context. We're going to get normal amounts of protein produced. And of course, we've converted what was previously background wild-type gene and protein into the mutant version as well. So from a model perspective, all these assays we can now perform, the model is far more representative. And I think this sentence really sums up why CRISPR-Cas9 has been so revolutionary, in that it allows us to build far better, more representative and accurate experimental models. And of course, any data we generate from these models will be far more relevant to the human disease. And this is a quick video that I have. I'm going to change the pointer back to normal so I can control the video. So you can see that's what Cas9 looks like. There's only two components, Cas9 and the guide RNA. And if I just pause the video here, oops, that failed. Let's just go back a second. So if we just pause the video a second, so that's what the Cas9 looks like, and it's coupled up to the guide RNA, just these two components. And the guide RNA, if we just pause the video in a second, hopefully it'll work this time. These nucleotides here, there's only 20 nucleotides just hanging out the edge here, but we can change those 20 nucleotides. Everything else, the Cas9 protein, the rest of the guide RNA sequence, it all remains the same. But we can change those 20 nucleotides and reprogram this complex to recognize different sequences of endogenous DNA. So if we fast forward this video now, what you'll see is when we put it into cell, it goes running up and down the genome, searching for a complementary site. And if it finds it, it can unwind the DNA at the endogenous location. And you get Watson-Crick base pairing of this guide RNA, those 20 nucleotides, to the endogenous DNA sequence here in the genome. And once it's there, the Cas9 nuclease activity becomes active. Well, why is that important? So in bacteria, this system is used as an antiviral adaptive immune system and degrades bacteria. So it's a nuclease to destroy DNA. When we put it in mammalian cells and human cells, it's able to cause a local bit of DNA damage. Now, of course, our DNA is being damaged all the time. We're exposed to ultraviolet radiation from sunlight, maybe some toxins that we consume in our food and drink. And when we get those DNA breaks, we've got a series of different DNA repair pathways that come into play. And that's all gene editing is. We're exploiting those repair pathways once we've deliberately made a double-strand break in the genome somewhere. So one of the main repair pathways we think about is non-homologous end joining. This is basically like a molecular glue. The cell recognizes the DNA has been broken, a bunch of enzymes come in, and they glue the DNA back together again. It's pretty good normally. It can make mistakes. Those mistakes typically are in the form of what we call indels, so small insertions or deletions. So it can be a bit imprecise at times. Alternatively, there is homology-directed repair, or HDR. Now, this occurs in a phase of the cell cycle where the cell knows that all its DNA has been replicated. So the cell knows that the broken DNA has got a perfect copy elsewhere. And those homologous regions can line up and it can copy that perfect copy to very, very precisely repair the broken DNA. We could subvert this process. We can hijack this process by supplying our own repair template with a little bit of homology flanking the region we want to change and enact a precise repair. So we exploit NHEJ with those two components, just the guide RNA and the Cas9, for gene knockouts. So you can imagine we make these indels, these small insertions and deletions, in the middle of a gene, then basically the gene stops functioning. If we supply a third component in the form of this donated DNA template, we can get very, very precise changes, like those gene knock-ins, like trying to make a point mutation cell line, that kind of thing. It is worthwhile pointing out that NHEJ is far more likely to occur in HDR, so that does present some challenges for us when we're trying to screen our cells or our mouse lines to find the ones that have got the change we want. I apologise in advance for the mildly patronising analogy I've got here, but if we imagine genes as being a sentence full of three-letter words, or those codons that form the amino acids, you can take a sentence like, she saw the big red fox eat the old fat hen, and let's imagine we took those molecular scissors and chopped out two letters. And if you look at the sentence at the bottom now, it's gibberish. It makes no sense anymore. And that's analogous to the gene knockouts. We're basically making genes gibberish within the cells, and they no longer produce the protein. With homology-directed repair, we add in that repair template. So here we've got the words red and the words the, which are homologous to this sentence, and they're flanking something to be changed. So if we cut here and HDR occurs, we change the sentence to, she saw the big red cat hug the old fat hen. We've still got a meaningful sentence, we've just deliberately changed the meaning. And that's analogous to when we may be making those point mutations in genes within cells. And that's how we can use these technologies in lots of different ways. And as I said, we tend to focus, our history has been mouse focused. I like this concept of the 4Ds for CRISPR. So no matter what model system you're working in, you can break it down into 4 different steps. There's the in silico step that starts with the design of the editing. So what does the guide RNA look like? What does the repair template look like? And then we move in vitro as we actually build the guide RNA, the Cas protein, and the repair template. Next, we move in vivo, where we deliver those reagents into, here we see a mouse embryo. And then finally, once we have pups born, so we'll then implant those embryos into a pseudo-pregnant female mouse. A mouse is born a few weeks later, and then we can take genomic DNA from those pups that are born, and we can try and detect the editing change, and that's performed ex vivo. It is worthwhile highlighting when we talk about animal models and using mice or zebrafish or another protected species, we actually had a 50 at the very start, which is discussion. And here we're discussing the ethical implications, what's the phenotype going to be like, and are we covered by our project license here at the University of Manchester to legally make this mouse model. So it's a really important step at the start when we're dealing with animal models. So I thought I'd run through an example of a mouse we've made previously, which harbored a mutation in the gene CSF1 to model this particular gene, this particular disease here, the human disease ALSP. And there's a mutation in these patients of the E633K, which basically mutates this tyrosine kinase domain of this receptor. Now, in the patients, it leads to neurodegeneration dementia, and one of the big characteristics is they lose these immune microglia cells from the brain. I don't want to get too much detail about the ins and outs of this, because in all honesty, I don't know much about the disease. We work with the research group, and they tell us about the disease and the mutations that they want to program in, and that's what we do. So we're not experts at the disease side of things. We're just building the models for them to work with. But essentially what happens is they end up with 3/4 of the receptors that have been formed by this dimerization not working properly. And when those receptors are not working properly, it means that we do know the patients no longer get the survival of these microglial cells within the brain. So how do we do this? So this is just zooming in on the exon in the mouse that contains glutamate 631. That's the residue that's analogous in mouse to the human. And you see here in blue, we've highlighted where the guide RNA is binding. So we've found a guide RNA that is very, very local to the site we want to change. And we've also built a repair template here, which is single-stranded DNA in this case. It's got 60 bases of homology on one side, 60 bases of homology on the other side. And you see here this bump in the middle where we're trying to change this codon of GAG to AAA. And if that change happens, it changes the glutamate to a lysine to model the disease. So that's the design aspect. Here, on this side, is the development of the reagents. When we first started doing this, 8 years ago now, time flies, I had to build everything myself in the laboratory. So synthesize the guide RNA, synthesize Cas9, synthesize the repair template. Whereas these days, I just buy it from a company. It's great. You know, one of the great things that's happened is commercial sector's really got on board with this technology, and it makes our life so much easier and allows us to streamline what we do. Sorry, 1 second. The video is not playing for some reason. So if we look at this video now, we load this injection needle here with those reagents. This holding needle holds the single cell mouse embryo in place, and you'll see the needle penetrates and injects the CRISPR-Cas9 and the repair template right into the pronucleus of this developing single cell mouse embryo. You know, people have been doing this since 1975, and we've just changed what's being delivered there. Now, of course, any genetic modification that we make at this stage of the mouse, as this embryo starts to divide and then differentiate into different organs and tissues, every single cell that results is going to harbour that same genetic change made at the one cell stage in the fully formed mouse. So it makes it super efficient for when we want to get germline transmission of this change to the next generation. And I don't want to labour the point about detection, because I'll be honest with you, it's probably the most boring aspect of the process. But we do a series of PCR assays and sequencing, that kind of thing. And then we identify this in pup number 16 from this one here. We've done an alignment. Allele number 1 is wild type. We've got the GAG sequence in there. Allele number 2 is the perfect sequence. And then we've established a colony, and we hand the mouse model over to the research group, who go away and do all those kind of assays and those type of things to really characterize the mouse and learn more about the human disease. And just as an endpoint to this particular story, we published this mouse model last year in the Journal of Development. And what you can see from these images down here, just to drive home the point, that in the wild-type mice, there's lots of microglial cells in the brain. In the dominant heterozygous mouse, we've got a reduced number of microglia in the brain. So now the research group have got a really useful tool to better understand the disease, and perhaps at one point in the future, maybe even use as a platform to try and develop therapeutics or things like that for this particular disease. As I say, we also use these same technologies in lots of other model systems, including culture cell lines. Again, I'm just going to throw a couple of examples out there. So, for example, these are K562 cells, where we've made this mutation, which is very common in leukemia, and it's in a gene called U2AF1, which is a splicing mutation. We've made them heterozygous, as Dan Wiseman, the PI, wanted. So you can see 50% of the alleles in this clonal population have got the mutant sequence, 50% are wild type, and now Dan's using this to do some transcriptomic analysis to ask the question, when we disrupt the spliceosome of these cells as associated with leukemia, what are the downstream impacts on gene expression? Does it give us something that maybe could be a candidate chemotherapeutic target in the future? We work very closely with Carl Cadler's lab, who studies fibrosis and collagen secretion, and we used CRISPR-Cas9 to put this fluorescent protein on collagens. So now the lab can see these collagen fibers being secreted from this cell here, and they can image this using confocal microscopy. And this is really important because fibrosis is a major disease issue in cancer, in post-surgery, and lots of different diseases. And we really don't understand very much how it works in vivo. So we've built an in vitro model in cell lines that allow us to put some quantification and measurements on collagen secretion and that kind of thing. And as a matter of fact, we've now recently built a mouse model with these same kind of tags, so we can do the same kind of thing in vivo as well. With Chiara's lab, we have made some breast cancer cells where we knocked out these receptors, which are critical drivers of some types of breast cancer. And Chiara's aim is to now use mass spec to understand how that changed the proteome of the cells, and again, what is involved in the signal transduction pathways from these receptors and the cell surface of breast cancer cells that make them into breast cancer cells. And again, can we uncover some new therapeutic targets there as well? And finally, this is just showing a time-lapse imaging. You don't really need to know too much. This is an old video of mine, but we're able to image multiple proteins in the same cell in an asynchronous population at the same time. You can see the red proteins going in and out of the nucleus, the green ones in the cytoplasm, but its levels are going up and down in different cell lines. So we can do some very, very high-end confocal time-lapse imaging experiments to understand how these the protein P65 and its inhibitor protein ACAP alpha, how they function in this cell line. And it's not just cell lines. We've worked with people in zebrafish as well. We work with Matthew Burkitt to make some embryonic stem cells that will report on how the stem cells differentiate into different mesoderm and endoderm endpoints. As a matter of fact, I mentioned the iPSC work we're currently trying to set up. We're working very closely with Matthew to do this as well. So he's bringing that expertise into the facility to help us establish that new service. And then I mentioned fruit flies as well. I have absolutely no idea really what shot one does at all. I just thought that Innis, who we work with, generated some very, very pretty images, and they were worthwhile showing, this shot one EGFP in the developing larvae of fruit flies. So that's the end of part one, and our kind of day-to-day existence, what we do all the time, and I've been doing for years and years and years. And you can see we use CRISPR to make improved models of disease in mouse cells and flies. We can knock genes out, we can make disease-relevant point mutations, we can tag genes, and we've made about 140 novel GM mouse models in the last 7 years. Since we started making cell lines, and admittedly we had a bit of a pandemic that got in the way when we're doing this, but we've been able to generate 120 completed cell projects in the last 3 years, 10 new fruit flies in the last year, and we've got a big bottleneck here with about 30 of these queued up right now. And we also clone lots and lots of plasmids for people too. I've not really gone into detail about this particular service, but as a service, we try and always integrate new things all the time to make sure the research colleagues we work with are getting the very best possible service. And that brings us to the next aspect. If we go look back at this workflow again about genetic disease, once we've done those assays, if something comes out to be diagnostically relevant and important, can we use CRISPR-Cas9 here as well? Let's go back to what I said at the start. We're using CRISPR-Cas9 and we're programming this guide RNA to recognize a specific DNA sequence within the cells. And once it gets in the cell, it binds the Cas9, drags the Cas9 to the target, breaks the DNA, and we exploit those repair pathways to make gene edits. Let's highlight that bit of the sentence there. Programmed to recognize a specific DNA sequence, which of course is the basis of many diagnostics. Maybe the type of things that you guys at EMQN are doing on a daily basis. So can we actually repurpose CRISPR as a diagnostic tool as well? Well, actually, where Cas9 is concerned, it's not very good here. It's great in cells, it does its job very, very well, but in a test tube for diagnostics, it's not particularly relevant. But CRISPR is a bacterial system, and there's lots of different species of bacteria out there. And what many groups have been doing is looking through those bacteria metagenomes and finding different systems, different Cas systems that have got different useful characteristics naturally. As an aside at the bottom here, there's lots and lots of people looking at bacterial immune systems these days because everyone's looking for the next CRISPR because it could be worth billions like CRISPR is. But just to highlight these two systems, Cas12 and Cas13, they function in very similar fashion to Cas9 in that you program a guide RNA to recognize a specific sequence. But then what happens afterwards? Cas12 targets the DNA and then has this indiscriminate DNA's activity. It just chops up random DNA around it. Cas13 is similar, although the difference here is it targets RNA rather than DNA. And this indiscriminate nuclease activity can be exploited. So this is what a CRISPR diagnostic assay might look like. Just put the points back up. And it only takes 4 components. So we've got the Cas13, we've got the guide RNA that we've programmed to recognize the target sequence. And in the test tube, we take these reporter probes. And these have got a quench molecule on one end and a fluorescence molecule on the other. And because of that quench molecule, we get no fluorescence. We add the RNA sample. And if that RNA sample contains a sequence recognized by CRISPR-Cas13 in this case, then it binds it and cuts it, and it goes crazy and starts cutting all these probes up as well, releasing the quench molecule away from the fluorescence molecule. And we can measure that fluorescence using one of these TCAM plate readers in the laboratory. And we've been applying this as an activity the last 18 months or so. It's a bit out of left field. It's kind of not disease modeling anymore, but it does play into our strengths with these kind of CRISPR systems. And initially, we were funding a project by the Defence Science and Technology Laboratory at Porton Down for pathogen diagnostics. So Elise from the lab, what she's been doing is designing these guide RNAs, or CRISPR RNAs as they're called here, up and down the genome of the pathogen, setting them off in this assay in a plate-based system, and then screening, saying, do any of these detect the pathogen sequence? And you can see that this is just 4 guide RNAs shown in one graph here, but this one didn't work at all, whereas these 2 were pretty good, and this one was really good. And just to highlight, it's quick. This is between 8 to 12 minutes or so, we're starting to get a fluorescent signal being detected here. So we can design guide RNAs to recognize specific pathogen sequences and just reprogram the system for different genetic targets. Those genetic targets may also include human genetic variants. So I understand that EMQN, I was told before this talk by Simon, that you are doing some quality assessment for some recently developed assays for the mitochondrial RNR1 variant. So, of course, this variant here, if people have got this variant and they get prescribed gentamicin as an antibiotic, it can actually lead to permanent hearing loss. About 1 in 400 to 500 people have that particular variant. So what we want to be able to do, if, say, an infant comes to clinic, diagnosed with sepsis, and you want to prescribe antibiotics, you want to make a very rapid decision, rapid choice. What antibiotics do you prescribe in this point of care setting? And in our fluorescence-based assay here, using admittedly synthetic RNA templates, so this is nice, clean RNA in a laboratory setting, we can see that the wild-type sequence with this guide RNA here gives us quite a strong fluorescence intensity, whereas if we plug in a mutant sequence, we get a very, very different fluorescence intensity. It still detects it, but at a far lower amount. So it is different to the pathogens. Pathogens is asking a yes-no question, whereas the genetic variants is asking a kind of some on less question. So it is a bit different, but we can, again, detect within 8 to 12 minutes or so a difference in fluorescence intensity. So this is very preliminary data we generated a few months ago. And we started to realise that, yeah, this is great. We can program these assays. We can build these assays. But this isn't fit for the clinic. This is a big fluorescent TCAM plate reader we've got in a laboratory. There's no way in the world you're going to ship a sample from the clinic to us to purify and set this assay and it work in a time perspective. So we recognise we need to be part of a team, an interdisciplinary team here. That involves the upstream part. As I mentioned, we're using synthetic templates, really nice and clean. That's not real world at all. We need to start think about what is going to be the patient sequence as well as the patient sample. How we work in saliva, how we work in blood, how we work in urine, we don't know. So what kind of preparation is going to be required? Do we maybe need to think about an amplification step if we're working with samples that generate only a very low amount of material? Then, of course, we can program this assay and screen guide RNAs and CRISPR RNAs to see if they work. But then the follow-on question is we have to ask the test. Where is the test taking place? What's the readout? So, of course, in fluorescence, it's laboratory compatible. But maybe we want to think about different outputs, like a lateral flow test, which would be compatible with maybe the field somewhere, or biosensors, which would be far more clinically compatible. So an interdisciplinary workflow requires an interdisciplinary team, and this is what we get. So we're calling ourselves the 4Cs collaboration, which stands for the CRISPR chemists and clinicians collaboration. I'm not personally a fan of the 4Cs as an acronym because I think it's open to abuse, but you can see this is the team we've assembled here with lots of different skill sets, and we've managed to attract an awful lot of funding as well, even from just a few months of activity. So as I mentioned, we're working with DSTL for detection of pathogens. We have got a position we're currently recruiting as part of the recently funded Manchester BRC, an experimental officer to do that kind of functional validation work of those variants of unknown significance. We got some money from the Advanced Biomaterials Centre here to fund Maddie Lombardo's PhD project. BBSC have funded us to develop a CRISPR-compatible biosensor based on Mike Turner's field effect transistor technology. Again, way out of my comfort zone and expertise, but can we repurpose the CRISPR diagnostic system into this point-of-care system? And finally, Bill and John led on this, the Devote application, which I understand the EMQN are part of, where we can develop a translational pipeline in collaboration with industry, universities, and healthcare professionals for time-critical genomic testing. I've mentioned RNR1, but there's other candidates as well, like CYP2C19 and CYP2D6, where we're going to plug them into this pipeline. And ultimately, I think that's what we're trying to establish, a plug-and-play workflow where new diagnostic assays can be tailored for genetic variants or pathogens of interest and tailored for the perspective of what's the sample, what's the sequence trying to detect, and what's the relevant readout we need to build as well. So that's the part 2 summary there. CRISPR can be used to develop programmable diagnostics. And for the last part of the talk, I'm going to talk about things we're not currently doing and the idea of using CRISPR in the clinic. So, of course, I talk about creating disease all the time. Why are we not thinking about using CRISPR to correct disease? Now, of course, that seems very plausible in principle, but in practice, it's not so easy. You know, we're talking about humans humans rather than cells in a dish. But we've seen some pioneering work on sickle cell disease in particular. So if you all cast your minds back to when you did your GCSEs, perhaps in the UK, we learned about hemoglobin and we learned about sickle cell disease. And it's the most common inherited blood disorder in the world, affecting millions of people. And I think one of the reasons why this has been a bit of a test bed for CRISPR in the clinic is the delivery aspect is easier because the cells are taken out of the patient, they're modified in a dish, and then engrafted back into the patient. So can we simply correct the mutation? So there's the patient, that's the mutation they've got, which causes sickle cell disease. If we use CRISPR-Cas9, can we correct that GTG to GAG, and that's going to go back in the bone marrow and give us healthy red blood cells. But remember what I said earlier on, HDR, precise editing, is far less likely to occur than NHEJ. And the reality is when this work started happening a few years ago, the percentage of correctly modified cells was too low to be clinically effective. So is there a way around this? And again, hopefully you all remember graphs like this, which highlight that we have multiple hemoglobin genes in our genomes. We have one called fetal hemoglobin, which is active in utero when we're not breathing for ourselves and we need to harvest oxygen from the maternal blood supply. So it's got a higher affinity for oxygen. Of course, when we're born, we don't need this anymore. So our genomes turn the hemoglobin F gene off after we're born. But of course, we've got a dormant hemoglobin gene that works in our genomes. If we could turn it back on again in the sickle cell patients, would that be therapeutically relevant? And this is where to highlight the interplay between basic research and clinical research, because decades of basic research identified how this gene turns off. And basically, in erythroid cells, a transcription factor called BCL11A is produced, it binds the gene and it turns it off. And in this paper here, which is pioneering this particular treatment, what they did was say, well, let's use CRISPR to disrupt the BCL11 gene and turn it off in these cells. So by hitting this enhancer, BCL11A is no longer turned on in red blood cells. If you don't have BCL11A, it can't bind to the fetal hemoglobin gene. It doesn't turn it off anymore, and the gene turns back on. So what I love about this is they leverage NHEJ as a repair mechanism that is therapeutically viable in this setting, rather than trying to get the precise HDR to work. And it's wonderful to start seeing some of the human faces behind these stories. So this is Victoria Gray, and this is an incredibly brave lady. She speaks at lots of conferences, a really inspirational speaker as well. And she suffered from sickle cell disease. She went through this process in 2019 to have her hematopoietic stem cells extracted, modified to disrupt that enhancer, and then put back into her body. And within weeks, she had no side effects. They could detect hemoglobin in the bloodstream, and her symptoms are gone. And you see, this is 2021, and I think this still stands today that she never had to go for an emergency room visit, blood transfusion, any kind of pain medication, that kind of thing. So her life has essentially been completely changed by this therapy, and it was a single treatment. It was essentially curative. So one treatment led to a lifelong treatment of her disease. I think I've tried to sum up here some of the challenges of getting CRISPR into the clinic. So we talked about specificity and precision. Obviously, in the case I said there, they could use NHEJ, and that's not going to be the case for every single disease out there. So we're looking at new solutions, including some derivative technologies, which I'll just mention in a second or two. Delivery is critically important here. Those cells were taken out of her body, edited in a dish. You can't do that with someone's liver, you can't do that with someone's brain, so you can't do it with someone's muscles. So for different diseases, we need to start thinking about different delivery modalities. And we're not only seeing viral vectors, which are very, very common, but things like lipid nanoparticles, which you'll all be familiar with from the COVID vaccines in the last few years. Virus-like particles as well. And this is a really hot area of research right now to start getting gene therapy products into people. There are also challenges that are not biological, such as health economics and ethical challenges. Just an example, this sickle cell treatment is being priced at roughly $2.5 to $3 million per patient. That's not going to be very accessible. It certainly will not be accessible for the vast majority of patients, even in higher income countries, and it certainly won't be accessible for people in low income countries as well. So the big question needs to be asked here, and I think the solution here is going to require an awful lot of political will and some big discussions going forward. $3 million a patient sounds like a lot of money. A sickle cell disease patient is estimated to cost most health systems $15 to $20 million over the course of their lifetime. So I would think about long-term benefits, or unfortunately, politicians often think in terms of short-termism instead, so short-term budgets. So this is a big question that needs addressing. And just to highlight this particular solution here with base editing, this is a derivative technology that improves the precision of CRISPR. It basically uses the CRISPR-Cas9 platform as a delivery system into the genome, and it blunts the scissors. It mutates Cas9 so it can no longer cut the DNA, but instead you attach an enzyme to it, like cytidine deaminase or adenine deaminase. And these enzymes directly convert base to DNA. So you can very, very precisely change DNA at the target site. And David Liu is an author on both these papers here. I should crop his name a little bit there. But this is generating a huge amount of excitement. And in some of the preclinical models you can see here, it rescued a mouse model of spinal muscular atrophy. It rescued a mouse model of progeria, the accelerated aging syndrome. And David Liu spun out a company called Beam Therapeutics, raised about $300 million. And they're looking at applying this technology in lots and lots of different settings. And again, a human face on this. This is really pioneering work, recently announced by Vaseem Kassim at Great Ormond Street Hospital in London. And he used base editing to support CAR T therapy for leukemia. So he took a donor T cell from another patient and used base editing to make alterations that meant that donor T cell would not attack the patient's own tissues. And then, added also the CAR T receptor in there as well. And this is Alyssa. She has, the BBC article from December highlights, incurable cancer. And it's 11 months in this article, we're going to be 18 months on now. She's in full remission following this treatment. So it's really exciting how we're seeing these diseases being treated in the clinic at the minute. There's a long way to go, but I think it's very exciting to see what's happening. And I think it's also worthwhile pointing out that as a facility who has expertise in these technologies, I think the future is very, very bright. What my intention is personally, is we should be aiming to maintain these services, keep doing what we do in these different model systems, but also join this developing ecosystem in Manchester, such as the BRC, the Rare Conditions Centre, these kind of things, to actually start thinking, can we bring in our expertise to start thinking about strategies that might contribute toward disease correction as well? And that's very much my intention for the next few years. Okay, so I'll summarise the talk there, say that gene editing is making its way into the clinic with really exciting preliminary results. And this is my team. So there's me on the left there, and this is Wei and Sam. These two do all our cell line engineering projects. Elisa pioneered our CRISPR diagnostics. Haley does all our molecular biology and cloning of lots of different vectors. And Adam and Ed, they run our mouse engineering pipeline as well. And as I highlighted, we're currently recruiting 3 new positions in the short term as well. So, thank you very much for your time today. I'm really happy to take any questions you might
have. Thank you so much, Anthony. That was a really very useful, logical and detailed explanation of CRISPR and its uses and how the technology is evolving. So thank you for that. We have had a couple of questions. I'm just going to reshare my screen.
Do you want to stop mine?
Yes, please. So now I've lost the questions.
I've got them. You want me to?
No, it's okay. I found them again. So I've just got quite a general question to start off with. So obviously it's quite a complex process to translate a research finding into something for diagnostic or clinical use. I'm just wondering if you could summarize the steps that are required to translate a proof of concept study into something that can be used in the clinic, and whether there are any major obstacles in that process.
So actually, no, I can't. I'm not best
Oh.
placed. Our expertise is very much at the basic research level, and what we do there. It's exactly why we've partnered with people like the clinicians like Bill Newman and John McDermott, who have got devices and tests into the clinic, and all those kind of accreditations you have to go through to say, is it as good as what's currently available? How fast is it? How accurate is it? How specific is it? This is really beyond our area of expertise. What we're very, very good at is design the proof of principle and screen the proof of principle, and then work in a collaborative fashion with the other experts. It's something, I mentioned as well, just as a wider point in science these days. There's so many technologies. There's so much to learn and understand, and that's why core facilities, I think, have evolved in the way they have, because it's unrealistic these days to expect a PhD student, and I can see Katie Sadler's asked a question there, who is a PhD student and know from the time in the lab. It's unrealistic to expect a PhD student to understand and learn every single one of these techniques and technologies and apply them to their maximum capacity. You have to have these core facilities and expert technologists in place to support the best possible research. So yeah, it's very much a team effort. And I think we're still learning with this process. Hopefully in a couple of years' time, I'll be able to answer that question for you more accurately.
I think we can summarize your answer with there's a clear need for different departments to collaborate more effectively, and I think that's going to be really important going forward. So we've got a question from Emma McCarthy, who is based in the Liverpool Genomics Laboratory, and she's asking, for the mitochondrial RNR1 work, does the assay handle different heteroplasmy levels? So kind of what's the limit of detection there?
I mean, that's a really good question, and that's something we're going to find out. We are cheating right now. We're using nice, clean, synthetic RNA samples. We're maybe spiking it into saliva and that kind of thing, but we don't actually know how that's going to work on real-world samples, and that's where Bill and John come in with providing those samples. There are things we can do in terms of amplification of those sequences as well prior to the assay that increase the sensitivity of the assay as well. But we don't know. Obviously, mitochondrial RNA. There was two reasons why we selected this as a initial pilot project, and one is there's definitely an unmet clinical need, and there's people working on this already. Number two is that it's a sample we can get from buccal swabs, and we're hoping that because it is a mitochondrial RNA, and we think it's very highly expressed, so it may be that we have sufficient levels of RNA to start with. I actually don't know how it's going to handle those kind of heteroplasm issues. That's something we will find out, as we work on this project.
So as you alluded to, Katie from the Manchester lab also asked the question. I don't know if she's a bit disgruntled from her PhD work. She's moved on now, but have you got prime editing working yet? She'd like to know.
So prime editing is another, just to let everyone else know, it's another derivative technology developed by David Liu, like base editing. Prime editing, again, it uses nuclease dead Cas9. It drags something else to the region of the genome in the form of reverse transcriptase. And then in the guide RNA, you add an RNA template that hopefully gets copied into the genome. So it allows you to not only do those point mutations, but it allows you to do small insertions and deletions precisely as well. I think it's fair to say it is very exciting in principle. In practice, it probably hasn't been as effective across the world in other people's laboratories as where it's initially developed. But this guy, David Liu, is absolutely incredible, and he is constantly evolving and improving these technologies. And some of the more recent papers that come out have really come up with later versions that are far more effective. And yes, we are looking at implementing prime editing as part of some research and development projects right now. Katie did try it as part of her PhD, and as I say, it wasn't particularly effective and particularly efficient, but it is an active area that we're working on at the minute.
I guess this is a good follow-on question there, because you just mentioned David Liu's work, but another question we've had is, how does CRISPR technology compare with the base editing technology from David Liu's lab at the Broad Institute? I don't know if you've already kind of answered that.
Kind of touched upon it. I mean, there are definitely stark differences between the two. So base editing, as great as it sounds, is a lot more restrictive in terms of where you can target the genome. The base editing window is really small. So if you don't have a viable guide RNA in the genome, you can't build a base editing method. And again, there are solutions being developed all the time, in particular by the Lou Lab, to address this. Base editing will not do every single base conversion that you want either. So you're limited to C to T or A to G. So there could be issues there as well that might not be relevant for your particular disease. Base editing is bigger. You not only got the Cas protein, but you've got the enzymes fused to it as well. So from a gene therapy perspective, you've got a much bigger genetic payload to get into the cells to work as well. So there are clear advantages of base editing, but again, there's clear advantages of classic CRISPR as well. Certainly from the point of view of what we're doing in the laboratory, 90% of the time we apply CRISPR, it is using CRISPR-Cas9, but we are starting to look at some projects where we say, actually, base editing might be the best way to go here. And that's something we're actively looking at across different projects. But yeah, it remains to be seen what's going to bear out. And I suspect it'll be a case of, for this particular disease, this particular variant, base editing is best. Whereas for this one, CRISPR-Cas9 is best.
Yeah. So we've got a question about kind of the efficiency of editing in mouse models. What do you find the editing efficiency is in mouse embryos? How many pups do you need to screen before you find one with the desired variant, or
it's
whatever
really
you're trying to make?
Really high efficiency.
Oh, right, okay.
So that aspects. If you think about cells in a dish, you've got to come up with a transfection method to get all the materials into the cells. Where we take a big sharp pointy needle and directly inject the CRISPR where it needs to be. And as a result of that, it's super efficient. We, generally speaking, don't make many knockout mice anymore. It's regarded as not being a particularly exciting mouse model these days. Because generally speaking, whenever we get pups born, every single one of them has got some form of editing in it. What we want is the precise editing. And even then, it's still pretty high. Some of the new techniques we're using, we typically get 50 to 70% knock-in efficiency. So it's really, really high. It's a very, very efficient process. From a 3Rs perspective, what that means is, when we first started using this technology, we might generate 30 pups per project. Now we aim to generate 10 at most, because there's no need to generate any more. We end up with multiple founders. So from a 3Rs perspective, we are reducing the number of mice we're generating. The counterbalance there is, because it is so efficient, the price comes down, the accessibility goes up, so we make more mouse models than ever before. So although we've reduced the number of mice we need on any particular project, we're doing more projects. So there's a bit of a counterbalance there. But yeah, it's really efficient in mouse embryo.
So we've got a couple more questions. Let me just check the time. Yeah, we've got time. So I think I might know your answer to this one, but we've got a participant who is wondering what kit you recommend to perform CRISPR editing to assess the effect of a variant of uncertain significance, or not kit, maybe chemistry, I don't know.
I mean, it's going to be completely dependent on what you're trying to look into. So if you're looking at a mutation in a transcription factor, for example, you might program mutation in, and then once you've defined the sequences in the cell, so we always do Sanger sequencing, we are moving to next-gen sequencing to see how efficient to detect the clones that have got the correct change. But you program mutation in, and then you ask the question, well, how does it change the transcriptome of those cells? What's the transcription have to bind to these days? Maybe do some kind of chip analysis to say, or chip-seq analysis, sorry, to say, where is it binding the genome? Is it targeting different gene targets? So you tailor it to the system. Maybe it's a cytokine that normally gets secreted. So if that mutation impairs secretion, or maybe it accelerates secretion, so you could use secretion-based assays. So that's really for the research group to do. We have a hand in that, because sometimes we help with assay development as well. But I say, to highlight, the system is reprogrammable, so we can tailor it to so many different disease settings and different model systems.
I guess this next question kind of follows on from that. So we have a participant who's wondering whether there are any protocols available to cut out specific regions of the genome, maybe by using two different Cas systems that target different sequences.
Yeah, you don't need to use different Cas systems. You stick with Cas9. two guide RNAs, so a guide RNA up here and a guide RNA down here, and you can actually delete everything in between. We successfully deleted 1/4 of a megabase from the mouse genome using this technique. So we had one guide RNA up here, and then 250 kilobases, another guide RNA here, and we actually did one of those really small single-stranded DNA repair templates, which had homology here, and seamlessly deleted everything in between really, really effectively. So yeah, we can use it for lots of different kind of genetic manipulations like that.
Great. And then we've got another question about the development of treatments for congenital adrenal hyperplasia. I'm not sure if you know anything about that, whether you've read anything about possibilities for treatment of metabolic disease, which is largely kind of enzyme replacement therapies. What do you know about that?
Oh, that's those 4Ds. Those 4Ds apply both in what we do in experimental models, and they apply in the clinic as well. So first of all, delivery. You need to have a platform that will efficiently deliver the gene editing reagents to the cells and tissues and organs that are affected by the disease. So there's a great example of transthyretin amyloidosis, which is a disease affected by mutations or a gene expressed in the liver. So the company doing that particular trial developed a lipid nanoparticle that was specifically programmed to be uptaken by hepatocytes. So again, I don't know this particular disease, the CAH, but if you know the cell types that are affected by that disease, can you come up with a delivery method to specifically target CRISPR to it? Once you've done that, you program the CRISPR that's been delivered, maybe it's base editing, maybe it's prime editing, maybe it's classic CRISPR-Cas9, to perhaps change or modify the enzyme to correct it. And again, the level of correction, how much is required? There's some really exciting things coming out in the field of Duchenne's muscular dystrophy, where we're seeing that you don't have to correct every single cell for it to be therapeutically viable. If you can correct 10% of the cells in the animal models, you actually produce enough protein now to stop the disease progressing. So it is a question of, how many cells do we need to target, and within those cells, how efficient does it have to be for it to be therapeutically viable? The sickle cell example, for example, those initial trials, they edited around about 30 to 40% of the cells effectively and put those back into the patient. So 60% of the cells were still sickle cell, but there are sufficient cells into the patient now that express beta hemoglobin for it to be therapeutically corrective.
So just to follow on from that then, to your knowledge, is a positive response to treatment with CRISPR-based therapeutics always guaranteed? And, are there any reported adverse side effects that you know about?
So my big concern is we will start to see adverse side effects. There's going to be a long follow-up on all these studies. The gene therapy field has had numerous setbacks in the past, such as the case of Jesse Geisinger in the 1990s, who at Penn State University, who died as a result of a reaction to the AAV delivery modality, not necessarily the gene therapy, the virus that delivered it. And tragically, there was very recently another person in a Duchenne muscular dystrophy trial who died as a result of an inappropriate immune response to the AAV that delivered the CRISPR into the person's body. It wasn't the CRISPR that killed the person, it was the delivery vector. I think that's why we're seeing a huge amount of work on LNPs, VLPs, things that are not going to be as reactive with our immune system. But yeah, it's going to depend on whether or not we can deliver effectively to those cell types. Also, in terms of long-term follow-up, I mentioned that example of the sickle cell disease. What if in that population of cells, there's a cell in there that got CRISPR modified in a really bad way that we didn't detect, and that's been put to the patient, and then in 10, 15 years' time, that cell develops in a nasty way, something like that. So we have to be really conscious that this is a possibility. And as we do with any kind of medical intervention, it's a cost-benefit ratio at play here. What is the risk-reward ratio, essentially? Was that both parts of your question? There's another part. I'm not sure I answered both parts there.
No, you did, definitely. Well, I asked if there's always a treatment response. I guess you did answer that already.
Yeah, I say, it's going to depend on the system. I mean, we're seeing people, the retina is another place where people are developing things because it's a closed immune system there. Things don't, generally speaking, leach out. So the retina is another place where we're seeing gene therapies, gene editing being applied in clinical trials.
So I think I'm just aware of the time, but I'll finish on a more positive question. So in your opinion, are the most exciting future uses of CRISPR?
I mean, it's got to be, the clinic essentially. We are this idea that we can do a single treatment to cure a patient of a lifelong genetic disease is so exciting. The sickle cell example. There's about 200 people walking around this planet right now who've been gene edited and gene edited successfully to cure them of various different diseases. The sickle cell treatment is worth our pointing out as well. Is actually a one size fits all for thalassemia as well, and we're starting to see that exact same treatment being used in thalassemia. It's worthwhile pointing out there are multiple companies that come up with slightly different gene editing approaches for sickle cell. So they'll be in competition with one another, which will hopefully lead to a price reduction. Ultimately, we live in a free market world, and that might be one of the benefits there. But I think, so the idea that we could do a single treatment to cure a person of a debilitating disease is very exciting. And there's a huge amount of growth in the rare disease area here, where people are actually funding and applying this in rare and ultra rare diseases as well. And that's going to inform all kinds of things. We can take generic findings from all those studies and apply them in different settings. So I think we're in a CRISPR revolution. I always say that. I think we're going to see an exponential increase in the number of people that are being successfully treated with this technology.
Great, thank you for such a thorough answer. So I think we'll end the Q&A session there. Thank you, Anthony, again for presenting for us today. It was really amazing. So I'd just like to let everybody know that our next webinar will be in June. The exact date is to be confirmed, but it will be from Alex Wagner, who is an assistant professor at the Institute of Genomic Medicine, Nationwide Children's Hospital in Ohio, and he's the director of the Variant Interpretation for Cancer Consortium, and he'll present on developing variant representation standards for scalable genomic medicine. So please look out for the registration emails from EMQN events. And we'll end the session there. Thank you, everybody. Thank you, Anthony, again. And I'll see you all soon. Thank you.